have a pointer here. All right. So James, thanks for inviting me to come and give this talk. And I have to admit that when you sent me the invitation, I was very busy, and I shot off this sort of ridiculously broad title, Modeling Chesapeake Bay. But that gives me a lot of leeway to talk about a lot of different things. But in my own defense, I will say that this year is my 20th year of doing modeling work in the Chesapeake Bay. So maybe I have some license to use a very broad title like this. But actually, perhaps a more accurate title would be something like this. What I'm really going to talk about today are some examples of some of the modeling-oriented research that I and my colleagues have done in Chesapeake Bay over the last 20 years. And I'm going to be delving into basically three very different modeling approaches, but all of which leverage hydrodynamic models, that is, simulation models of the flow of the Chesapeake. And I'm going to be talking about some individual-based modeling approaches, and I assume that most of you, I'm sure most of you here have some sense of what I mean when I say that. I'm going to be talking about some of the work that we've done in terms of ecological forecasting in the Chesapeake Bay, trying to predict distributions and occurrences of species. And then finally, I'm going to wrap it up, and I'm going to talk about an ongoing project on modeling hypoxia in Chesapeake Bay, which is perhaps the most relevant to the theme of this meeting because it's a very data-intensive project. But there is a theme that runs through all of this work, and it's the notion of using three-dimensional, time-dependent hydrodynamic models to provide insight into biogeochemical and ecological processes in Chesapeake Bay. So all of these efforts are oriented toward achieving this goal. So let's start with individual-based modeling. So as an overarching objective, our goal is to simulate the impact of three-dimensional currents and mixing on pelagic organisms in Chesapeake Bay and how these interact with behavior to determine the fate of these organisms. And I'm going to show you two studies that I've been involved in. The first is a rather old paper called Modeling Particles and Pelagic Organisms in Chesapeake Bay, Convergent Features Control Plankton Distributions. But I want to start with this because this was really the first modeling study in the Chesapeake Bay that brought these types of IBM-type modeling approaches to look at these kinds of questions with pelagic organisms. And it really started it opened up a whole bunch of work, much of it led by my former postdoc and now colleague Elizabeth North. And I'm going to talk about, I'm also going to show you very briefly as another case study, this paper called Modeling the Influence of Episodic Events on Transport of Striped Bass Eggs to the Estuarine Turbidity Maximum Nursery Area. Okay? And this work, Elizabeth has really taken off with this work and has really become sort of the guru of individual-based modeling. And you may be familiar, she's published an open source model called LTrans, which is a general tool now for, I believe it's a systems model. And this is all this work that I'm going to talk about has been funded by the National Science Foundation. So, if you know anything about individual-based modeling, you know that it's fundamentally really simple. And I'm talking about individual-based modeling in a three-dimensional flow field. Okay, so that's the context. Okay, and the basic approach is that you can model the movement of a particle or an organism, and consider that movement in a hydrodynamic flow field as being a combination of a large-scale deterministic velocity vector, and that's code word for meaning that's the part of the flow we can actually model with these rather coarse resolution hydrodynamic models, combined with a small-scale turbulent velocity vector, which is really what we're doing here is we're parameterizing the effect of turbulence on these organisms. Okay, and then of course you can finite difference this equation, and you can say, say, well, at each time step we can move, and you can use a modeled large-scale flow field, and we can add in a parameterized turbulent kick, and we can move a particle around in a three-dimensional flow field. And then that's great. But the really interesting stuff starts to happen when you start to add in things like behavior, sinking, and behavior. Okay, and so looking at this very early work that I did in my 1999 paper. What we did was we took an early version of the granddaddy Chesapeake Bay hydrodynamic model. It's called CH3D, curvilinear hydrodynamics in 3 dimensions. This is a kind of a low resolution version of it, and this provides us with that deterministic large-scale velocity field that we can model. And then what we do in this particular study is we use a correlated random walk model to specify the turbulent part of the movement. And this particular study was a 2-dimensional study, okay? And this shows an example of the residual surface flow for average in July for the Chesapeake Bay from that model, okay? It's a 2-dimensional large-scale deterministic velocity field for a 2-dimensional application where we're going to be moving particles around in the surface. So there's an implicit assumption that these are buoyant particles that stay at the surface, okay? And of course, to do this type of modeling, what we're going to be doing in this study, what we did was we continuously added these particles, like dropping them onto the surface of the Chesapeake Bay and let the currents move them around, okay? And we kept adding them and adding them and adding them, and they can't sink, as you'll see in a minute, it turns out that they reveal some things about the Chesapeake that we didn't realize before. And one of the things I want to point out to you here is that if you look at this residual flow field, it's the average, so it's averaging over many, many tidal cycles for a month. You'll note that there's this rather dense velocity region here that in the lower bay, and it turns out that this modeling simulation revealed a residual eddy in the lower Chesapeake Bay that we didn't actually know existed before the study, and it was completely serendipitous. We weren't looking for it. And what we found out is that when you continuously add these particles to the surface of the Chesapeake Bay, they accumulate in areas where there's downwelling circulations. Okay, of course, that's what they're going to do. These particles can't go down, so if the flow goes down consistently in a particular spot, they're going to accumulate in that spot. And we discovered that there's this area of the lower bay where we have this residual downwelling eddy. It's called the Chesapeake Bay Accumulation Zone, and what we found is that these particles go into this region and get trapped there, that's neat. Nobody had ever seen that before, and it turned out that when we started looking at, I was involved in a big survey project at that time called the TIES project. They were measuring everything they could over the entire Chesapeake biologically. And it turned out that when you started looking at some of the other biogeochemical and biological fields, that this residual feature that we revealed by the model shows up rather strikingly in some of the biological fields. And this just shows that in the summertime, you very often in satellite data will see enhanced chlorophyll concentrations in this residual eddy domain. You also can see jellyfish accumulations. These are Hydrodromedusii. These are little tiny jellyfish in the Chesapeake Bay, which show pronounced enhances in their concentration in this residual eddy. And you even see accumulations of bay anchovy, probably a behavioral response. It's probably that these little fish are swimming to that region because there's a lot of food there. And we also know, based upon benthic studies, that this is a region of exceptionally high benthic productivity in Chesapeake Bay because stuff is coming there, it's getting trapped, and ultimately raining out and feeding the benthos. So anyway, that was the early study that started a lot of subsequent work. And so now what I want to do is talk just briefly about this second IBM modeling study. It was published in 2 papers by Elizabeth, myself, and colleagues in 2005 and 2006, and it's called, as I said, Modeling the Influence of Episodic Events on Transport of Striped Bass Eggs to the ETM. And so, of course, I got to explain what an ETM is. An ETM, that's the estuarine turbidity maximum, and it turns out in most estuaries that the upper at the head of the estuary where the salt water comes in, and these are isohalines, where the salt water comes in and comes in the bay and meets the outgoing fresh water, which comes up and flows over the top. it creates a trapping zone and a recirculation, and also a highly turbid zone. Okay, and this occurs in, as I said, in most estuaries. And it turns out that this iconic fish, the striped bass, a very important recreational fish in the Chesapeake Bay, that this is a really important nursery area for these fishes. So, in this study, and this is a three-dimensional study as opposed to a two-dimensional study, but we're using the same methodologies where we're finite differencing this equation, we're using models to specify a large-scale deterministic velocity vector, sort of the larger scale flow, and then we're also putting in a small-scale turbulent velocity vector. But in this case, we're being a little bit more sophisticated, and we're using these models, these hydrodynamic models, not only calculate the flow, but they also calculate turbulence. And we're using the turbulence specified by the turbulence closure models in these hydrodynamic models to scale this parameterized turbulence in the three-dimensional flow. And so we can finite difference that. And then what we're going to do here is that these eggs, these stripe-backed eggs, they are slightly negatively buoyant. They sink a little bit. Okay, they're a little bit heavier than the water, and that turns out to be a really important reason why they end up getting trapped in this ETM, in this nursery area in the Chesapeake Bay. And this is it was an idealized study. We weren't using a realistic model, and this looks like a really funny model, but I'm showing you here is this is the extended outer domain of the model, which has nothing to do with what we're simulating, other than we're using the cardinal rule of hydrodynamic modeling is to try to get your boundary as far away as possible from the area of interest. So we extended it 2,000 kilometers out, but the area of interest is only this little 98 kilometer section, which is where we're simulating the estuarine, the turbidity maximum in an idealized fashion. Okay. And this is a little bit more of a complicated problem because we're doing it in 3 dimensions, so you have to interpolate not only horizontally in space to the particle positions, but you also have to interpolate vertically. And so we are interpolating the velocities vertically to the particle positions, and we're also interpolating the vertical diffusivity profile to scale the turbulence in the model so that we're giving these little particles little random kicks. As I said, and we're also adding sinking, and we did experiments with having different densities of eggs to see how the specific gravity of the eggs affected their trapping in the estuarine turbidity maximum zone. And these are the basic simulation results. And let me explain what we're plotting here. We're plotting over 100 kilometers. It's a little hard for me to see this. And depth, the Chesapeake Bay is pretty shallow, so we're only going down to a little over 10 meters. And what you're seeing here is the saltwater wedge coming up into the Chesapeake, right? And then the fresh water flows over the top. And then this gray stuff is we had a Eulerian model embedded in this to simulate the turbidity as an Eulerian tracer. And so, as you can see, we simulate a very nice turbidity maximum. And what these plots show is that when you get the egg density just right, actually realistic to how dense these striped bass eggs are, they tend to trap in the model right in the ETM. They tend to sink down and sit in there, and they recycle, they go around in that recirculation. If they're too light, they blow out. Okay, they just go right over the top and blow out. If they're too heavy, they get stuck like a little clot on the bottom. Okay, so it turns out that if you actually specify realistic egg densities, you get the right answer. You get the trapping of these eggs, of these striped bass eggs. It also turns out that if you have a big wind event, we simulated wind events will have a tendency to blow those eggs out of the ETM. Okay, so if you get a big wind pulse, it can just blast the whole population out of the ETM. And if they're really light, they really get blasted out of the ETM. And if they're heavy, they're just stuck on the bottom. And it also turns out that if you have a river pulse, they can also tend to push the eggs out. It's a little hard to see here, but what's happening here is that if you have a strong river pulse, it's tending to suck those eggs out over the top of the right onto the picnic line and out of the bay, and then you can see the light eggs getting flushed out as well. So it turns out that we gained some pretty important insight that the spawning after river flow events may promote that it's best for striped bass eggs, for striped bass to release their eggs after wind events and river flow events. And it's best because that way they don't get blown out of the ETM, but it's also a time after these river flow events, you get a lot of nutrients and stuff coming into the bay, and so there's a lot of food there. And in fact, these fishes tend to time their egg releases to after these events, so it optimizes their retention and the food available to them. So that's the North et al. Study. And as I said, Elizabeth is continuing on. In fact, we have a brand new grant that we just got from the National Science Foundation, a Coastal Seas grant, to apply this to oyster larvae for management applications. So now we're going to shift gear and talk about something quite different, but again using the same underlying hydrodynamic engines that drive the models that we use, and I'm going to talk about ecological forecasting in Chesapeake Bay. And I think hopefully that term means something to you. The idea being, as I said before, that we can use combinations of mechanistic hydrodynamic models with empirical models of species to try to predict the occurrence of species and where they're likely to occur in the Chesapeake Bay. So our overarching objective, and this is ongoing work funded by NOAA, is to provide nowcasts and short-term forecasts, like weather forecasts, of a variety of different types of organisms in the Chesapeake Bay. And at present, we can do forecasting, nowcasting and forecasting, just like the weather for sea nettle, that's a jellyfish, a stinging jellyfish, some harmful algal bloom species, a couple of pathogens, and we can also do physical and biogeochemical weather. We can model in real time the physics and the biogeochemistry in the Chesapeake Bay, and these are used for a variety of things: research, management, and public uses in the Chesapeake. And the approach that we use for all of this stuff is that we use, as I said, a mechanistic hydrodynamic and biogeochemical models to force data-driven empirical models that predict the likelihood of the presence of an organism in the Chesapeake. And this is based upon many years of work, starting back in 2006, our first sea nettle model, and is ongoing in the present. And so the hydrodynamic engine that we use for this work is called ChesROMS. Oops. Wow, how'd that happen? Wow, that was amazing. There we go. The hydrodynamic engine is ChesROMS. It's a three-dimensional hydrodynamic model of the Chesapeake Bay. It's an implementation of the Regional Ocean Modeling System. Our current version, which is available through systems, is a little bit older version of ROMS. We are working on updating that to the latest version of ROMS at this time. It includes all the major tributaries, and we've doing a lot of not only hindcasting work, but the key point here is that we have operational implementations. We actually have operational implementations that is a model that's run every day, and every day provides a nowcast and a short-term forecast. We run it at my laboratory at the University of Maryland Center for Environmental Science, and it's also being run with a different name at NOAA. It's called CBOS, the CBOS model, okay? And this is also open source. It's available through systems, but also through SourceForge. So this is the engine. This is how we generate the three-dimensional temperature and salinity fields, and also we have an embedded biogeochemical model, so we can nowcast and forecast biogeochemical fields. But the point of this talk I want to emphasize is the ecological forecasting. That is predicting species. And what we've done is we've developed a suite of empirical models, data-driven empirical models, for predicting things like sea nettles, these stinging jellyfish, and that's an empirical logistic regression model based on model-predicted TNS. We can also do some HAB species, for example, Carlobidinium veneficum. That's actually a neural network model based on temperature and salinity and time of year. And we also can simulate two species of Vibrio, the famous Vibrio cholera, and also a less famous but actually more concerning Vibrio vulnificus in the Chesapeake Bay. And these are also both logistic regression models based upon that are driven by hydrodynamically modeled temperature and salinity fields. And so just to give you a sense of the way it works, it's very simple. And this is an example for both sea nettles and Vibrio volnificus, and it turns out that the sea nettle, which is the species name, if you're interested, is Chryseora quinquecira, can become very abundant in the Chesapeake Bay during the summer months, and they sting people, so people care about that. In addition, Vibrio volnificus, human pathogen, also becomes very abundant during the summer, and it's a significant potential human health threat. And by coincidence, or maybe not by coincidence, we don't really understand it, temperature and salinity strongly controls the presence of both of these organisms, very different organisms, obviously. But it turns out that, for example, this is data, I hope you can see this, for the sea nettle. This is sea nettle abundance up to 20 per cubic meter, 20 per cubic meter. We get a lot of jellyfish in the Chesapeake Bay, okay? Versus salinity, versus temperature. And as you can see here, these really high densities occur in a very narrow, generally in a very narrow salinity regime, about 10 to 15 salinity, and they occur only in very warm water, okay, up above 20 degrees. Okay, and it turns out, by coincidence or not, that Vibrio volnificus, this human pathogen, has a very similar habitat, a very similar dependence on TNS. So it turns out that in the Chesapeake Bay, when you're getting stung by jellyfish, you're also exposing yourself to a rather nasty human pathogen. But we actually don't release that information, so people don't know you're also in a bath of pathogens when they're around the sea nettles. But basically the way this model works is we have a empirical logistic regression model that relates temperature and salinity to probability of encounter. And then what we do is with our hydrodynamics, we can predict the temperature, we can predict the salinity, those feed into the regression model, and they give us a prediction of the probability of encounter of these organisms. And both of these models are quite robust. For the sea nettle, it's robust because these sea nettles have really strong, especially for salinities, they have strong osmotic constraints, and they can only live in a very specific salinity regime. And they also, for complicated reasons that we basically understand, have a strong temperature dependence. So that's how the model works. And what we do is we generate maps daily. Of sea nettles for the Chesapeake Bay. We have a weather forecast for jellyfish in the Chesapeake Bay that's posted every day. We also have maps for volnificus, but they are not posted publicly because it's a very sensitive product. They may be someday in the future. They're used now for management applications. We provide nowcasts, so you can say, well, what's the probability? If I go out to a certain spot in the Chesapeake Bay today, what's the probability that I'm going to get stung by a jellyfish? And we provide 3-day forecasts, so you can look and say, well, where should I go this weekend to go swimming if I want to avoid getting stung by a jellyfish? Just for this example, this is an example of a nowcast forecast that I pulled last week on the 18th. And as you can see, and what we're seeing now as the water warms, the probabilities are increasing for encountering both sea nettles and volnificus. It's still a little bit too cold. As you can see also that the high probabilities occur in these mesohaline regions, about 10 to 15 salinity units, and you can see that those are that those mesohaline regions are basically occurring right now in the upper Chesapeake and in sort of the middle tributaries, and that's because we've had a rather dry month, and what happens in the Chesapeake when it's very dry is that the salinity, the freshwater flow drops, and the salinities gradually they migrate upriver, up bay. And so this is the current situation. It may change dramatically as we come into the summer season. We'll see. So anyway, that's how that works. So let's move on to the last part of the talk. Modeling hypoxia in Chesapeake Bay. And as I said, this is perhaps the most relevant part of this presentation to the theme of this meeting, because we're using a lot of data to validate our models, and we're also doing model comparisons on comparing models, how well they can simulate hypoxia in Chesapeake Bay. And the objective of this project is to assess the readiness and maturity of a suite of existing estuarine community models for determining past, present, and future hypoxia events within the Chesapeake Bay in order to accelerate the transition of hypoxia model formulations and products from academic research to operational centers. So it's the same idea as a sea nettle model. We're trying to test out and explore different types of models for modeling hypoxia to see which ones work the best for putting them in operational mode, so we can move toward having oxygen weather forecast for the Chesapeake Bay, okay? And we're very close to it, actually. The capability is just around the corner. There's really two fundamental questions that we're asking in this project, and that is that we can basically break down these hypoxia models into really simple one-term constant respiration models versus full biogeochemical models, and we want to compare these types of formulations to see how well simple models work compared to more complicated models, and then we want to figure out if we can use these for operational applications. And it turns out that in the Chesapeake Bay, rather surprisingly, traditionally for modeling hypoxia and oxygen, you would think about running a full water quality model, right? That incorporates the nutrient loads and calculates how everything's cycling and organic matter production and ultimately drawdown of oxygen. But it turns out that you can simulate oxygen drawdown in the Chesapeake Bay remarkably well by simply specifying a constant respiration rate in the Chesapeake Bay, constant in time, constant in space, and it turns out that the physical mixing and advection in the bay will set up a remarkably realistic hypoxic zone and anoxic zones in the Chesapeake, indicating that physics is really important in driving hypoxia. But so we're contrasting this incredibly simple approach of just specifying a constant respiration versus a fully dynamic biogeochemical models. We're actually testing several of them that respond not only to the physics like the simple model, but also to nutrient forcing. Okay. And what we want to know is, well, how well does this simple model work, and why do we care about that? We care about that because it's really easy to operationalize simple models. It's hard to operationalize complicated models, okay? So that's really what it resolves around. We can operationalize this really easily, and we want to know how well it works. And so we're working at two different time scales. That's intraseasonal, that is within season on weeks to months. So how well do these models work within a season? And we're also interested in the larger question of how well will these models work over interannual time scales. And for the intraseasonal comparisons, we're comparing the output, and I'm not going to try not to bore you with this, but we're comparing the output from 6 Chesapeake Bay models. We're focusing right now on 2004 and 2005. And to make a long story short, there's basically, as you can see, the Chesroms engine is being used in here extensively. And these 3 models are 3 full complicated biogeochemical models, but they're different. Okay, some are more complicated than the others. And then we're using 2 different models, a Chesroms 1 versus CBOS 1, that are running this real simple one-term model. Okay, and we're also comparing this to the granddaddy EPA Chesapeake Bay biologically sophisticated model called CH3-ICM. This is the model that we use for doing water quality simulation and setting TMDLs, total maximum daily loads, in Chesapeake Bay. Okay, and what we're interested in doing is examining how well they reproduce the mean and the spatial and seasonal variability of temperature, salinity, stratification, and these things all contribute to dissolved oxygen, as well as for the more complex models, the simple ones don't have this, chlorophyll A and nitrate, okay? And the real target is to see how well these models reproduce dissolved oxygen, but we also are looking at the drivers. These are the drivers. These are the physical drivers and the biological drivers that result in oxygen drawdown. And it's a very data-intensive study. We're comparing simulations from all of these multiple models, not only to one another, but to observations over 10 main stem stations for 16 cruises in 2004 and 2005, and these dots indicate where those stations are. And I have to explain this plot that we use. We're using what's called a target diagram for our skill assessment. Not all of it, but a big chunk of it. And how many people have seen a target diagram before? Can you raise your hand if you know, if you're familiar with it? Okay, so not very many. Okay, so what this diagram does is it expresses for a particular data point as compared to the model, and so depending on where this data point, when you compare the model to the data, where it lies in here tells you first how well the model simulation of this parameter, let's call it temperature, is modeling the real temperature. And it turns out that if for this method, if it lies on this circle, it means the model skill in modeling temperature is the same as if you just specified the mean temperature. Okay, so if you just took all of the data and averaged it, it means that the model result is only as good as the mean, so which isn't very good. And so what you really want is your model to be better than the mean. You want it to be simulating variability inside this mean circle, but it also provides a whole bunch of other important information. If it turns out if your dot is over on this side of the center is perfect. If your dot is here, it means your model's perfect. You get a perfect simulation of you're perfectly reproducing the observations. If it's over here on the right, it means you're overestimating the variability or the standard deviation. So if that dot falls over here, you've got too much variability in your model. If it falls over here, you have too little. Okay? And if that dot falls above the center, it means that you have overestimated the mean. You are biased high. And if the dot falls below, you are biased low, okay? And so, for example, this dot shows that your model is overestimating the variability, right? It's on the right side out toward this area. You've got too much variability, but you're biased low, okay? And you're not doing nearly as well. You'd be better off just using the mean because your model isn't near as good as the mean. So that's how it works. That's how that plot works. I know that's a lot of information in a short period of time, but I'm only going to show you one plot. And this is a summary of a whole bunch of model simulations for a variety of parameters, including temperature, salinity, stratification, chlorophyll, bottom nitrate, and bottom DO. And to make a very long story short, what we're finding in these model intercomparisons, and we're also comparing this in terms of both time and space, we find that these hydrodynamic models, and we're testing multiple hydrodynamics, that they all have good high skill in terms of bottom temperature and salinity. We're interested in the bottom particularly because that's where the hypoxia is, but they also do pretty well. Wow, how did it do that? Gee whiz. I'm pushing a button that I don't know what it is. Anyway, okay, so all of these models have fairly high skill in terms of bottom temperature and salinity, and that's the red and the yellow. So you can see that the reds and the yellows are generally, this outer ring is the mean, okay? So we're doing better than the mean in terms of temperature and salinity, but it turns out that they don't do very well in terms of stratification, chlorophyll, and nitrate. These, what would we consider to be strong determinants of DO, okay? And those are expressed by stratification is blue, surface chlorophyll is this sort of pink, and this aqua is the bottom nitrate. And you can see that the blues and the pinks and the aquas generally fall, not all of them, but so we're not doing very well in terms of stratification and chlorophyll, but remarkably, the models still get the high, they actually simulate DO with fairly high skill, and the bottom DO is the green one, and you can see that the little green circles tend to fall closer to the center. So even though we don't reproduce stratification and these biological drivers very well, the models still reproduce the bottom DO fairly well, for reasons which I can explain to you if you're interested. We actually understand that. And another interesting result that comes out of this is that it turns out that these really simple constant respiration models work as well as the more complex models. And these two stars are the real simple respiration models. And as you can see, for bottom DO, those stars are right in the center there. So these simple models work just as well. I'm almost done, just like last slide. Work just as well as these super complicated models. And so we also are now just beginning the process looking at interannual variability, and these are comparisons of 20 years of simulation of hypoxia versus time using one-term respiration model versus hypoxia versus time using the full a full complex. This is the CH3D ICM water quality EPA model. And to make a long story short, it turns out that at least in terms of the preliminary results, that these simple respiration models actually reproduce the hypoxic volume variability better than the fully complex biogeochemical models. So anyway, that brings me to the end. I'll just put those up for you to read if you fell asleep during my talk, and leave it there.
There were lots of interesting things in your talk. I'll talk about something at the end. The modeling intercomparison that you did was really nice, and the conclusion about the simple models. How is that being received? And
very badly.
Especially to this group.
Our friends and colleagues at the APA hate it because they've invested so many years in developing this model called ICM. And the verdict's not totally in yet, but our preliminary results are tending to show that the simple respiration models work better than the more complex models. And that's counterintuitive, of course, because they don't incorporate nutrient loading. The simple models have no response to nutrient loading variability. And my own take on that is that it may be related to our ability to tune these complex models, okay? The respiration model is really simple. It's one value. You can run your model over and over and over again until you twiddle that thing until you get it just right, okay, with one value. Whereas, as you know, when you run a full biogeochemical model, you might have 25 state variables, 30 state and that many or twice that many parameters. And so it may just be an optimization issue. In other words, that we don't have the computational resources to run, or the time to run these complicated models over and over and over and over again, or maybe the mental capacity. If you ever tried to tune one of these models, it's really tough. And that may be that simple.
And in judging it, are you judging it based, when you talk about skill, there's doing what you said and tuning the model and getting a good match, and then there's predictive capability,
right,
which is very different. And you're
yes,
judging this on predictive capability?
Not yet.
Oh.
We're only looking at...
And it doesn't really count in some ways yet.
Well, what counts, one take-home message is that even when you tune your model to the data and you look at the skill of what you're tuning to, the models still really struggle. Even when you're tuning to the data and not withholding data. When you start talking about withholding data and assessing the skill versus data that you haven't used to tune your model, all bets are off. Okay, so I think the message here is actually still a very strong message. It means even when we're doing everything we can to get the model to work with the data at hand, they still don't work very well.
That's a complicated thing. Yeah, it'll be interesting to talk.
Do you think the comparison that your simple model can predict even better than the complicated model, is that just for the Chesapeake Bay, or have you applied this, do this comparison for other sites or large scale comparison? I mean, it could be because the condition of that bay, maybe stratification is not that important, so tend to be more spatial or uniform.
That's an excellent point. And yes, no, we have not made these types of comparisons for other systems, but your point is very well taken, because the Chesapeake Bay is really strongly forced, atmospherically forced. Okay, we have a huge amount of atmospheric forcing variability, and as you point out, that if you went to a system that was perhaps not as strongly physically forced, this conclusion would not hold, and that the nutrient forcing would be more important, and you would need to have models that respond to nutrient forcing. That's a very good point. Thank you, Raleigh. You're welcome.
So those of us who like to write complicated models, we have lots to think about now. Okay, we're going to take a break now, but when we come back, you'll be coming back to different rooms. And where's Lauren? I think this is how it's going, that on the back of your card, you should, your tag, there should be a room that outlines what your breakout group should be. If it says 1.1, that's in the North Bay, and the North Bay is this one. And if it says 1.2, that's the South Bay on the other side of this wall. If it says 1.3, you're in room 2126, up a set of stairs. You've got 2 sets of stairs to get there. 2503 is also upstairs, and that's for breakout group 1.4, and the lobby is for 1.5. You just stay out there after the break and finish up the food. No, I mean you meet and you have your discussion out there. So let's take our break, and you know where to come back. Okay, thank you.